Happy Ganesh Chaturthi for everyone. So in this video we will discuss what are amino acids, right? We all know, we all have basic idea about amino acids, that the amino acids are some molecule which have carbon, hydrogen, oxygen, nitrogen and sometimes even sulfur also. So basically if you'll see one amino acid, it will have at least one alpha carbon, one carboxylic acid group and one amino group. Okay, one hydrogen will be present at third position of this alpha carbon, and then at fourth position any kind of functional group is possible. So acid means carboxylic acid, amino acid. Okay, so this is what basic structure of any amino acid. Like you see, as for example, you see this one amino acid, that is glycine. Okay, yes, yeah, see, at body pH the glycine is present in this form only. That's why many places I have made it positive, NH2 group as a positively charged and COH group is negatively charged, right? So this is, this is glycine. Actually glycine have, you see, carboxylic acid group present, right, and amino group present, hydrogen present, and at functional group hydrogen present, right? So this is what our glycine is called as simplest amino acid. Okay, when this in functional group hydrogen is replaced by one methyl group, we'll call it alanine. So alanine is another type of amino acid. All right, many example we have that we will see one by one in this video. Now you see this unique amino acid proline. See, this proline amino acid, is unique amino acid which have not amino group. Yes, which have not amino group not present primarily here. Actually they have one carboxylic acid group and then they have one NH group. Yes, what present, students? They have one NH group present. Yes, NH group present, and one that NH group also present in one ring. That ring is called imidazole ring, right? So this amino acid have not amino group, this amino acid have imino group, right? At body pH, at body pH, this carboxylic acid will lose its hydrogen, right? And this NH group, nitrogen have lone pair, that will accept one more hydrogen and they will become NH2 like that. So that's why initially I made it in ionized form. But I just want to say here that in proline amino acid, imidazole ring present, right? Imino group present, primary amino acid not present here, but NH, that means secondary amino acid present, right? Now if we'll see the types of amino acid can classify with by different basis. Like a very important basis of classification of amino acid is that standard and non-standard amino acid. See, amino acids, if you'll see, these amino acids are act as a monomer for protein synthesis, isn't it or not? Proteins are huge molecule which have thousands of amino acids, isn't it or not? When thousands of amino acids linked with each other by peptide bond, they form protein. Now this protein synthesis in cell is done by our ribosomes. So the ribosomes, if they are the amino acid, in nature we have hundreds of amino acids, right? But in that 100 of amino acids, only 22 amino acids added by ribosome in protein synthesis. They used to incorporate in protein synthesis. Now the amino acid which used to incorporate in protein synthesis by ribosome are called standard amino acid, and the like glycine, alanine, proline and all. and the amino acid which not incorporate by ribosome during protein synthesis are called non-standard amino acid. Like you'll see these two amino acid, ornithine and citrulline, they have amino group also, they have carboxylic acid group also, but they do not added by our amino acids during protein synthesis. Actually this ornithine and citrulline, is intermediate of urea cycle. During urea formation in our liver cells, ornithine and citrulline amino acid form, right? Apart from that, we have other example of non-standard amino acid like n-formyl methionine, which formed by our methionine amino acid, then desmosine formed by lysine, then 5 hydroxy lysine present formed by lysine amino acid, 5,4-hydroxyproline formed by proline. Actually these all amino acids, they are present in protein. Yes, they are present in protein. Like this 4-hydroxyproline and 5-hydroxylysine they are present in protein. Actually, see, this amino acid formed by modification in standard amino acid. Generally, like lysine was added in protein, right? And after protein synthesis, some changes was introduced in that protein, and then we converted into the 5-hydroxylysine. Like proline was added in protein, after protein synthesis some changes was introduced into the proline and it became 4-hydroxylysine. So these all amino acid, even they are non-standard amino acid, still they are present in protein. They are added in protein post translationally. Post translationally means after protein synthesis. Okay, translation means protein synthesis, we will see later, right? So they are added in protein synthesis later. After protein synthesis they are added in protein by the they formed in protein by modification in standard amino acid. Now come to standard amino acid. See, all standard amino acids are, they are alpha amino acid, and in alpha amino acid are, they are L alpha amino acids. All standard amino acid, we have 22 standard amino acid, that all are L alpha amino acid. Now you may ask, sir, what is this L amino acid and D amino acid? You see, this is one amino acid, suppose, right? It's alanine. Now you see the orientation of amino group here is present left hand side or right hand side. It's present left hand side, so we will call it L amino acid. Okay, yeah, like glucose, L glucose and D glucose, right? Now you see here amino group present at left hand side or right hand side? It's present right hand side, so we will call it D amino acids. These D amino acids are, rare in nature. Okay, many places they are present, like they are present into the cell wall of the bacteria, that is different thing, but these all standard amino acids are L amino acids, L alpha amino acid. Okay, this is carboxylic acid group. On this carboxylic acid group, this is what our alpha carbon. On this alpha carbon only amino group present, that's why we are calling it alpha amino acid, right? Now what are that 22 standard amino acid? Let's see, it's very interesting. We have again classified these all standard amino acid on the basis of their chemical nature. Like amino acid which have non-polar side chain. Non-polar side chain, obviously they will be hydrophobic in nature. They are less soluble in water. So what are that example? Glycine, which is simplest amino acid. Alanine, which have methyl group as a side chain, right? Valine, you see, hydrophobic amino acid. Leucine, you see, hydrophobic amino acid. Isoleucine, you see, these all are example of hydrophobic amino acid. They are actually, least soluble in water. Yes, they are actually least soluble in water. Okay, next one is our aromatic amino acids. If you'll see, aromatic amino acid means they have aromatic ring present, isn't it, students? They have aromatic ring actually. Like you see phenylalanine, tryptophan and tyrosine. Phenylalanine, you see here, it have also one ring. Then tryptophan, you see tryptophan have also ring. And then you see tyrosine amino acid, tyrosine amino acid have also ring, right? So unique, so because they have aromatic ring, that's why we are calling them aromatic amino acid. The aromatic amino acid have one unique ability, they can absorb ultraviolet rays. Yeah, they can absorb ultraviolet rays. And average value, if you'll see, they can absorb maximum light at 280 nanometer. Like you see our phenylalanine absorb light, maximum light at 260 nanometer. I'm writing here 260 nanometer nearby. Tryptophan absorb light at 280 nanometer, right? And our tyrosine and tryptophan absorb maximum light at 280 nanometer, right? So I just want to say they have aromatic ring, and in this aromatic ring, you see pi electrons are present. The pi electrons have ability to absorb light, and that they absorb light at different wavelength, but they absorb maximum light at 260 nanometer. I mean the average value will see it's a 280 nanometer. Yes. So these all are example of aromatic amino acid. And then we have some more amino acids, like we have proline, we saw. Then methionine, you see methionine is sulfur containing amino acid. One second, methionine I have not drawn here. Yes, this is our methionine. Sulfur, it have sulfur containing amino acid. Two amino acids are sulfur containing amino acid. One amino acid is methionine. Okay, it have also non-polar side chain. And second one is our sulfhydryl group containing amino acid, that is cysteine. Cysteine is example of sulfur group containing amino acid. You see here sulfhydryl group present here, sulfur group containing amino acid. Now third amino acid we classified uncharged polar side chain. Uncharged polar side chain means, if you'll see our physiological pH, body pH nearby 7.4, you will see, or average value when you will see 7 also, or 7.4 better you see in context of physiological pH. So at physiological pH, this amino acid side chain is uncharged. Side chain means, this also R group, they are uncharged. So serine, you see OH group containing amino acid. Threonine, you see threonine is also OH group containing amino acid. Cysteine, you see SH group containing amino acid. Asparagine, you see it have amide group. Yeah, CONH2 present, you see. So asparagine have amide group. And then we have glutamine, you see glutamine have also even amide group. What is difference between this glutamine and asparagine? Yes, amino group here also, carboxylic acid group here also, hydrogen here also. Amino acid here also, hydrogen here also. But in side chain, you see 2 carbon and 1 amide group present. Here only 1 carbon and then amide group present, right? And then our tyrosine amino acid. Tyrosine we have written it's already, right? Okay, tyrosine I have made it 2 times. Tyrosine is aromatic amino acid. One side it's aromatic amino acid, another side is it have uncharged polar side chain at physiological pH 7.4, or you 7, you say same thing only. Okay, so aromatic amino acids, aromatic amino, you'll see, except the phenylalanine and tryptophan, they have non-polar side chain, right? But tyrosine have uncharged polar side chain. Fine. Is it clear, students? Okay. Now you see next one, very important. Amino acid with charged polar side chain. That means at pH 7 or physiological pH is 7.4, right? So at physiological pH, their side chains are charged. Like if we'll see these three amino acid, arginine, lysine, histidine. Arginine, lysine, histidine are also called basic amino acid. Basic means alkaline wala basic amino acids. Why we are calling it basic amino acid? In their side chain you see amino group present or not? Yeah, in their side chain amino group present. Yes. So apart from one amino group, they have some extra amino group present in their side chain, right? That's why sometimes we are also calling them monocarboxylic diamino acid. Why? Because they have only one carboxylic group but more than one amino group present, right? So arginine, lysine, histidine are example of basic amino acid, right? And then aspartate and glutamate. Aspartate and glutamate are example of acidic amino acid because you see in their side chain also carboxylic acid group present. Yes. So aspartate and glutamate are acidic amino acid. They are acidic amino acids are negatively charged at pH 7. At pH 7 you will see they are negatively charged, or pH 7.4 they are negatively charged. Arginine, lysine, histidine are positively charged at pH 7. Question is that how? Question is that why? We will see that all things in next video, right? In this video what we saw? In this video we saw that two amino acid, they have amide group and they are glutamine and asparagine. Yes, asparagine and glutamine have amide group. See, amide group present. This is asparagine and then we have glutamate amide group, right? Our arginine, lysine and histidine have extra amino group present, more than one amino group present. Sulfur containing amino acid are what, student? Cysteine is sulfur containing amino acid, methionine is sulfur containing amino acid, right? And then we saw two amino acid which have extra carboxylic acid group. Aspartate and glutamate have extra carboxylic acid group. And then we saw hydroxyl group. Serine and threonine have extra hydroxyl group. Yes, serine and threonine have extra hydroxyl group. One second. This is serine, this is threonine. They have actually hydroxyl group, right? Hydroxyl group containing amino acid there. And then we saw aromatic amino acid, tyrosine, phenylalanine, tryptophan. Phenylalanine, tryptophan, tyrosine are aromatic amino acid, but in this aromatic amino acid, phenylalanine and tryptophan, these two are they have non-polar side chain, right? But tyrosine have uncharged polar side chain. When their side chain, tyrosine side chain is polar, but at pH 7 they are uncharged, right? And then we discussed that our aromatic amino acid have unique ability to absorb light. Okay, phenylalanine absorb maximum light at 260 nanometer, tryptophan absorb maximum light at 280 nanometer, and tyrosine absorb maximum light at 280 nanometer. Okay, about pyrrolysine and selenocysteine I have made separate video. Remaining we'll see in next video. Tata, bye.